Well, good morning, everyone. To you and to the people who is watching online again. I'm afraid that I was not very responsive to the people who was online because the chat thing. So I will try to monitor the chat. And again, if there are any questions, maybe live, if you see some questions in the chat, just let me know. Okay, so let us continue. Thanks again for coming at this time. So I retitled these lectures in this way. So we are talking about dynamics of biological systems, but in particular, I don't know why, because I got this thing. This must be from some other. No, this is okay. Okay. So, so the type of biological systems that we are considering here are gene regulatory circuits. I defined more or less what we consider to be a circuit yesterday, and I would like to continue with this today. So it's going to be very basic what I'm going to be talking about, already introducing mathematical approaches to this. So the math is, my work I can define as doing very simple math on very complicated problems. But since what I do is the modeling, I don't think it has a lot of merit, but the problems are really, okay. There is some feedback in live. Okay, so anyway, let me remind you the plan. And I am, as expected, very delayed. So these things, maybe I will shorten this, and I don't think it's a big deal because this is not very a core issue of the dynamical systems community. So I am not going to be very specific about this. I will just talk a bit about the phenomenology and some of the approaches, but I'm not going to go too much into the details in point 3. This allows me to continue going, even though I'm a little bit delayed with the plan. So we are still here. Okay, and as usual, let me remind you, I haven't emailed anything to Josep because hopefully you have access to this thing. And then I have already posted the slides from yesterday, not the ones from today. So the idea is to continue talking about the dynamics of gene circuits. And let me remind you what we discussed yesterday very briefly. This is what I claim to be the challenges of the field that we are calling systems biology. And I'm using this, I haven't defined it, but anyway, it's the same idea. A dynamical system is a system, and if you apply this approach to biology, this is what I call systems biology, which is very different from what other people call systems biology, which is omics and lots of data and so forth. And I think these other people, this other philosophy of systems biology is somehow winning a bit, again, due to the advantage, to the arrival of these omics procedures. But I'm keeping small in that way. Okay, so anyway, these are the challenges of this field of systems biology, and some of them. And what I was saying is that molecular interactions are usually nonlinear, are highly abundant. There are many of them, many degrees of freedom, and also important for us is that they're strongly dynamic. So I think we are here because especially of challenges 1 and 3. And I would like to talk, well, sorry, I would like to talk now a little bit about this challenge number 2 again, going back to challenge number 2. But as I say, challenges 1 and 3 is what brings us together, in my opinion. Okay. Now, I just want to remind you of this paradigm that Tyson and Novak proposed, which I think is very clear. And the idea is to use this type of bifurcation theory approaches, which you can do in vivo. And that's one of the things that I would like to show either later today or tomorrow. But you can do bifurcation analysis in vivo, in the real cells, and you can do it, of course, in your computer, and then you can compare. And from this comparison, you learn something. That's the approach. That's this dynamical paradigm. So these guys were amazing in phrasing something that it's very clear something that in which I have been working for years. This is my philosophy, but I never was able to put it as nicely as in this figure here. Anyway, the problem with this is with challenge number 2, because if I say that there are many, many interactions and many, many components, this means that the dimensionality of the dynamical systems are very large, and I don't need to tell you. Then the bifurcation analysis become very complicated. We know very well how to do one-dimensional bifurcation analysis or 2D. We can look at codimension 2 bifurcations, and then it becomes very complicated in our minds to interpret. So we still have a problem coming from the fact that we have a lot of components. So this is, for instance, a circuit, a gene circuit, that we compiled this many years ago about the circadian clock in Drosophila, in the fruit fly, okay? This was the state of the art, if you will, in 2007. Since then, of course, many more components are found because people find that when the fly or the biological organism lacks one particular gene, the rhythms are altered, and then immediately this gene is put in the map. And then, of course, other people now, they investigate how they interact with all the other elements, and then you have something like this. Now, this is not the largest one, but I just wanted to point out, I mean, imagine this is a gene circuit that it's already sizable. So you have 20 dimensions. You have a lot of parameters, as we were saying the other day, this curse of dimensionality, if you will. But my point is that we cannot apply really this thing to something as complicated as this, in my opinion, because doing the dynamical systems approach is complicated with so many degrees of freedom. Okay. Now, luckily, we have math to help us, because if we have a clock, we have a circadian clock. This is a clock. Okay. So we have 20 or whatever the differential equations that are going to go into a limit cycle. But of course, we know from dynamical systems theory that we do not need 20 degrees of freedom to generate an oscillator, right? We only need 2. If there is delay in the interactions, we only need 1, but I'm not going to enter into delays. Okay? Delay differential equations. I'm not going to go into this. If there is no delay and interactions are instantaneous, with only 2 degrees, and I'm forgetting about noise and so forth, anyway, but we know that with 2 degrees of freedom, we get limit cycles. You know, Imma has explained, and I'm sure that Thomas will continue talking about this today. So I think this is very fitting. I'm using this slide, by the way, in my talks to biologists, still the same. I don't touch it. And I think, and in that case, I have to tell them, please trust me, because they don't, it's unclear to them about these pictures, but for you, it's very evident. And I'm going to use the opportunity to just show you this slide and say, okay, we only need 2 proteins or 2 genes to generate a clock. Already we have 3. We were talking about this yesterday. We can have chaotic behavior. So I am convinced that there is a lot of, I am personally convinced. And again, this goes in YouTube and we will see, but I'm personally convinced that there is chaotic behavior in cells. It's just that we haven't found it because it's difficult to distinguish from noise, but I'm convinced there's no other way. It's either noise dominating everything or really deterministic dynamics dominating everything. And with 3 degrees of freedom, we have chaos and nonlinearities. Come on, okay? And a lot of people, I think, Blay included, and the group of Ricard Soler and Josep, and we have been looking for examples that are clear, and probably Thomas, and we haven't found this, but I'm not going to talk about this right now, but this is another point to discuss in the future. Okay, I think this is a hole in our understanding, because chaotic dynamics and strange attractors are very important in terms of the ability of controlling them, of predicting the future, of synchronizing. So it is very important if we can identify this type of dynamics. These are complicated and they have high capability of information processing, and yet you can control them, synchronize them, and predict them. But I'm going to focus right now on this, if you will. In the plane, you can have interesting dynamics with only 2 degrees of freedom. And now I go back to the biological system. I think, look, okay, evolution is not wasteful somehow. I mean, biological systems are great optimizers. No, so evolution is going to really force you to optimize, because if you're not optimal, you're producing you have to produce lots of genes to generate a behavior that you can produce with 1 gene less. I would hope that a mutation will come, and a circuit with 1 gene less is going to be more effective. You don't need so much energy to maintain it. So the cell with 1 gene less, as long as they can oscillate, it's going to work. So eventually, I cannot believe that you need all of these genes to oscillate. Of course, people working in circadian biology and doing thesis on each one of these arrows are going to be disappointed. I don't want to make them such. I'm not going to say that all these interactions are not important, but there has to be cores. There has to be core circuits with smaller degrees of freedom that are responsible for this dynamical behavior that is so important that we saw yesterday. So this is one of my motifs, one of my thoughts that I always have, okay? That there are inside this large, and this is not very large in comparison. I mean, I'm not talking here about 30,000 or 20,000 degrees of freedom. I'm just talking about 20, but I think that we don't even need to go to 20. There has to be smaller modules, okay? And that's the whole idea. So how do we prove that? So One way of proving this experimentally, or at least testing this hypothesis experimentally, is with synthetic biology. Now, this is cool. This is cool about synthetic biology, and I have been working a lot about this. So I would like to claim that we can prove that with only 2 or a smaller number of degrees of freedom, you can get limit cycles. And you can do that by saying, okay, I'm going to set up a small gene circuits like this. And I know that I can analyze them. I can write down the differential equations using the techniques that I'm going to be discussing in a few minutes. So I'm again jumping ahead. But I can model the dynamics of these circuits with ODEs. And then I can look whether I find limit cycle behaviors or not. Of course, we expect them to be there because we know that nonlinear dynamical systems with 2 or 3 degrees of freedom are going to oscillate. So, but in this case, you don't even need math. It's intuitive, if you will, because look, if I have a gene A whose product is repressing the production of gene B, whose product is repressing the production of gene C, whose product is repressing the production of gene A, what's happening? If I start with high A, B is low. If B is low, C is high. If C is high, A is low. Definitely frustration here. So the only way out of this is, well, one of the way outs is that you get an oscillation. And the same thing here. A is high, and this is expressing a repressor. This is an activator that activates a repressor. Okay, so if A is high, then R is high, and then A has to be low. So there has to be regions in parameter space in which this thing has to oscillate. And indeed, this is the case. You can analyze them, and there are lots of works about this. You can put it in a computer. I'm not putting the equations, but you can see just by doing simulations that this can oscillate in relatively large regions of parameter space. So in my opinion, that's one of the uses of this type of approaches. So here we have mathematical results. Now you can introduce these circuits using classical, more or less classical techniques of genetic engineering, cutting and pasting genes, and put these circuits inside the cell. Okay, and then what these circuits are doing is they are using the genetic machinery of the cells. They are using all the enzymes, the ribosomes, the RNA polymerases, everything. So they are just using the cell. And ideally, these proteins are not interacting with the rest of the machinery. So they are isolated. Of course, they are never completely isolated, but we assume that these genes are completely isolated with the other 20,000 or 30,000 genes and the million proteins. And then we test whether this circuit is going to lead to oscillations. And this was done 20 years ago. And this, in my opinion, initiated the field of synthetic biology seriously. This is the first example. And I think you can see here, these are Escherichia coli cells. These are bacteria that we have in our guts, non-pathogenic in general. So these are workhorses of molecular biology. A lot of people know how to use them. It's easy to do molecular biology and introduce the circuits here. And you can see that this is fluorescence, going back to the fluorescent proteins I was telling you yesterday. And you can see that the cells are blinking in this production of one of the proteins, one of the 3 proteins here. So these cells are clocks. They are not very good clocks. We know that this is not a good clock, and I'm sure that you know better than I. So engineers don't consider this a good clock. This one is way better. And this is not exactly the same. It has an extra loop here, a negative. But anyway, this is a more recent experiment in which you see the cells blinking. If you follow one of the cells, you see it blinking. Okay. Again, it's not perfect. It's noise. There is noise, but it works. So for me, this is a demonstration that you do not need large numbers of degrees of freedom to generate dynamics. You use synthetic biology for that purpose. Okay. So this means that around that time, so the idea was in the air. 2 years before this paper was published, there was this paper in, where was it, in Nature, at the end of the millennium or whatever, by big names. These are all big names, PIs in their own labs, well, big names. John Hopfield, I don't need to tell you, Stan Leibler, who was working with Michaelis in this repressilator thing that I just showed you, Andrew Murray, and Lena Harwer. So these are big names, and they got together and they write an opinion paper. And they call this From Molecular to Modular Cell Biology. This is a figure for the paper, and the claim is that now, at that time, they thought, oh, cell biology is maybe modular because we have modules like a radio has modules, and then you have here the controller of the volume, here you have the sensor of the detector of the signal and whatever, and the tuner. So we have modules. So that's the idea. For us, having a module means that we have circuits with a small number of degrees of freedom, and that's what I would like to claim, that we can still use the tools of dynamical systems theory in 2 or 3 dimensions, or 1 or whatever, to learn about real biology, because in some cases, this is modular. Now, of course, we have advanced a lot in 20 years. Somebody might claim we haven't really seen proof. I think for me, the proof is in synthetic biology. And I'm going to show you examples of us later today or tomorrow, in which we have one example in which we have a module. And we have been testing this example for years, and it works. And there are others, of course. So in other words, that's it. Let me go back. So that's a typical example. Here, 2 degrees of freedom. Well, there are subtleties here I'm not going to enter, because you might say that you might need more than 2 because you have the mRNAs that I'm not putting here. So there might be 4, but I'm not going to enter into this right now. I think we will discuss about this later, okay? So there can be 4 or 2. But anyway, the question is, how do we model these things? And now I also tell this to biologists. For you, maybe we can go faster. Each one of these arrows is a term in a differential equation. So the only thing that we have to do, we can translate directly this arrow diagram into 2 differential equations in which each arrow is a term. So now the question is, how do we make the translation? So that's the next step. Now we are going to enter into math a little bit. It's going to be very simple. I apologize in advance, but anyway, I think this is better, and that's what I know. So I don't know things as complicated as Inman, Thomas, and Bayra. So well, I'm just saying. So I'm doing things very simple. So first of all, let me start with a very simple process. This is protein dilution. This diagram is this is not even a paper. This is some kind of that is available in the internet. Nitz and Rosenfeld was at that time in the group of Uri alone. I don't know where he is right now, but he wrote this thing which is called a math primer for the perplexed gene expressionist, and it's a very nice introduction of these mathematical issues for biologists. And they were using these kind of things, but I think it's useful for us also. So I imagine that I want to model protein dilution. What does it mean? So first of all, this is a cell, a bacterium in principle. You see that they are rod-like. This is very simple. There's no nucleus, so it's not a complicated object. It's like a small bioreactor with stuff, so enzymes that will make a production of protein possible. Now, I am going to assume that there is no production. The red things here are copies of a given protein, and there's no production of this protein anymore. These proteins were there because they were produced earlier, and now there are ways experimentally to say, I'm going to stop production of this protein, okay? So now there are always 8, because I'm not generating, hopefully, I'm not generating more, okay? And this happens all the time. At some point, one cell decides not to express the gene anymore. So the protein the numbers are not changing. But of course, the cells are proliferating. Bacteria, if you give them food, they're extremely good at just proliferating. And they proliferate exponentially, right? Because, let me put it here. The number of cells grows exponentially. I am going to assume infinite current capacity. I'm not entering I don't need to model logistic growth here. I mean, eventually, of course, the bacteria are going to hit that limit, and we can see this in typical growth curves of bacteria, but I'm forgetting about this right now. I'm assuming infinite amount of nutrients, so they grow exponentially, okay? So of course, when they grow exponentially, their number doubles every time and again, but of course, the 8 proteins are shared. Eventually, you have none. Well, you always have, in this particular case, I'm assuming that these proteins are stable. We'll come back to the stability, to the finite stability, but if they are infinitely stable, some cells are always having 1, but most of the cells are 0. The protein is diluted. Okay, so this is called protein dilution. It's a standard way in which you are going to lose proteins. If the cells are proliferating, such as bacteria or the cells in our epithelium or whatever, they're going to lose concentration of protein due to dilution. Now, another thing that will happen, of course, is that these things are not infinitely stable. We are at room temperature. These are very big objects that are being hit by many other things. Eventually, they're going to lose their shape. And when they lose their shape, they are not useful anymore. We cannot recognize them as our proteins. They are something else. So these things are going to disappear spontaneously. Also, not spontaneously, you can also have enzymes. We saw yesterday the movie in which you have a protease. There are enzymes that eat up proteins. So this disappearance can also be produced by other proteins, which makes it even more interesting. I mean, biological systems are so amazing. So anyway, so this can also happen due to degradation. Okay, so now how do we model this? Well, it's very simple. The concentration of the protein will be the mass of the molecule divided by the number of copies of the cell. Since the n goes exponentially, this decays exponentially. So in a differential equation form, you just have this. So whenever you want to model protein dilution or protein degradation, in general, protein decay, That's a term. So that's not saying anything new. I'm sorry, that's why I was saying that this is very simple. But for biologists, it's very simple. It's very important because I'll tell you, look, if you want to model dilution, you just put a term like this in your differential equation of all the proteins that are going to be losing concentration for whatever reason. Okay? And of course, this coefficient here is related in this particular case with the cell cycle time in this very simple manner. So if we measure the cell cycle time, then we can measure this alpha, whatever. Now, what happens if we also produce, now imagine that the gene is turned on and is producing protein at a constant rate. This is what biologists call constitutive expression or constitutive protein production. For me, I don't know, that's my simplistic view. This means constant production at a constant rate. So for instance, in this case, I would just add to the equation that has the decay term, just a constant, and that's the simplest equation that tells me how much concentration, how is the protein concentration going to vary. We have a linear equation. We have to balance production and loss. We put this in a, we do a graphical analysis, for instance. That's what I like to do with biology because you don't need to solve. Of course, you can solve this equation analytically. It's linear. But since anticipating that later I'm going to introduce nonlinearities and I'm going to not be able to solve them, I just tell them, look, you can do it graphically. Just plot the phase line, plot the function f, and of course this takes you to the fixed point, which in this particular case is stable because the f is positive here, negative here, so it's an attractor clearly. And of course you can do the stability as Imma told you on Monday and mathematically. So in this case, in our case, it's not a problem. But I tell biologists, you don't even need to do this. You just need to know how to plot graphs. Okay? So that's the first thing I wanted to tell you. Now, maybe, shall we go to clickers or maybe not? Are you bored? Do you want to do it or do you think it's silly? Come on, somebody tell me something. We can skip it. Oh, okay, so let's do it. So now, so those of you who are in the online, just do you go to the Plickers website, my.plickers.com. You have it, you see it here. So then what I'm going to do, is I'm going to change today. I think this will work better. So let me see if this works. Okay, so now I am going to play this. I think this is going to work. I'm learning. Yesterday was the first day. It was a bit. So, question. If you don't have your card, you can download the Plickers card. If you didn't bring it, just download it in your cell phone and show me the cell phone. It also works. Okay. So anyway, I'm ready. I see that Abel is online. So Abel was you are online. No, you are not. Sorry. Okay. Okay, Damian already answered. Somebody who wants to answer something? Carlo? Okay, I'm taking Abel's, Lorenzo, Martin. Oh, sorry, there's not enough. Oh, it's too far away. Let me just get closer. There's a limitation. This is probably my camera. Marta, Julia, Natalie, and Julia. Okay. Okay, I'm sorry that we don't have paper. Anyway, so I think are we all? Is somebody missing? There's not so many people. I feel sad. Anyway, shall we stop? Damian already answered. Okay, so let's look at the answers. Wow. Okay, so show graph. Most people answer A. Okay, which maybe I did it incorrectly. Let me see. Which of the following statement is correct regarding a gene that is expressed constitutively? Most of you said that the concentration goes to 0 if the decay term is larger than the production term, but non-zero, none of them, because, well, maybe you'll tell me no, but you didn't express, so anyway, we can discuss this. The system can never exhibit genetic oscillations, and both of the statements, so anyway, so, okay, what do I do? So let me go to this. Let's see if we can have a discussion, if you will, now. Come on. So where do we go? Here. Okay, so point number one, I was saying the concentration is going to go to 0. The idea would be, oh, there is a balance and maybe if production wins over decay, we are going to go to 0. But that's not true in this case, unless the production term was equal to 0. So if somebody answered because he was thinking 0 is smaller than, no, we should have said, I apologize. If I'm always going to have some production, because I haven't told you this, but there is always some. If you have a gene in a cell, there's always going to be some production, because just by chance the polymerase and all the factors maybe go there. It's leaky. Promoters are very frequently leaky. I mean, zero doesn't exist in real life, so that's why. So if the production term is non-zero, it is not true that there is a balance, and maybe the error comes from just thinking in terms of exponential growth. And I think Thomas was introducing this equation the other day, in which you have dn dt. Oh, sorry, no, it's okay. But imagine if it's exponential and you have dn dt equal to alpha minus beta n. Of course, depending on whether alpha is larger than beta, you can go to 0. But I just wanted to point out this is not the case here, because the production is constant. Okay, so it's a very simple thing that you're always going to have a non-zero fixed point. Okay, it can be small, but it's always going to be non-zero. It's just there is always a non-zero fixed point in this thing. And what I tell my biology students is, look, I just draw a line and I know immediately what's going to happen. This system can only go to one point, to one fixed point, and stay there forever. If you start with no protein expressed, you're going to increase up to here. If you start with a lot of protein, you're going to decrease up to here, and that's it. Okay, so I wouldn't claim that A was correct. So I apologize if I misled you. Point number 2, this system, I was saying, can never obtain genetic oscillations, and that is true. As I was saying earlier, I'm forgetting about delays, because this is a ordinary differential equation system, not a delay differential equation. So I mean, if I am here, I can only go up. So this means that I cannot oscillate. I need 2 degrees of freedom. With only 1 degree of freedom, I either go, given if I am in this point of the phase line, I'm always going up, and here I'm always going down. To oscillate, I need to be able to go up and back and forth, and I need 2 degrees of freedom. So the correct answer was B in the thing. Okay, sorry, and this is, oh my goodness, I'm being too thin. Okay, so the correct answer was this one. Okay, I hope it is okay. Okay, so now I am going to stop this and I'm going to go back to the presentation. I hope it was not too disturbing or anything. So let's continue. Now, this is very trivial. It's linear, it's a linear differential equation. You are completely bored. Now, let's make it just a little bit more interesting, just by remembering that, of course, as I think it was Stefano, who is not, I don't know if he's here, I don't want to track people, sorry, I just feel like, I'm sorry. But I think it was Stefano who was saying yesterday that, no, you have to regulate expression, right? So this means that now the production, sometimes it will not be constant. It will depend on other proteins or on the protein itself. So how do you model gene regulation? Okay. How do you model the fact that you have transcription factors that control expression of the gene, and this thing is not necessarily linear, very frequently it's not linear, okay? So anyway, now I'm not sure if this is helps. I have to be honest with you, these are slides that I use for my biologist colleagues, students. And in biology, chemistry, and even in physics, they tell you all the time about enzyme kinetics. The only math, most of the times, the only math they have seen in their degree is enzyme kinetics. That I don't know if you have seen this in any part of your training, and I'm not going to go into this. Is the so-called Michaelis-Menten Kinetics, in which you have an enzyme. The enzyme binds a substrate. The enzyme is a catalyzer of a chemical reaction. The chemical reaction is to go from the substrate to the product. So of course you could go directly, and it will take forever. But if you have an enzyme that catalyzes a reaction, it binds to the substrate, but reversibly you have a complex, and the complex, in this particular case, it unidirectionally produces the product and releases the enzyme again, so the enzyme is conserved. This kind of dynamics is everywhere in all the textbooks in enzyme kinetics, and I'm telling you, this is one of the few things that many biology students ever see mathematically, but I do see the math. Okay, so since they are so familiar, I use it to model this regulation, because I can map with these reactions exactly what's happening more or less in gene regulation. If E now is the promoter of the, sorry, I'm introducing new terminology. This, what I call DNA promoter, I don't have it here. I think I do have it. sorry, can I do a detour? Let me do a detour. I have here some slides that I prepared. I was sad that I was not going to use, but I'm going to use now. Okay. Okay. I was telling you yesterday that we have a gene, and this little arrow was just an indication that the gene is there. It's going to be produced. It's going to be translated in. But this gene is how do you may ask how the gene how the expression of the gene is controlled. There is a little bit of a. I'm oversimplifying here. There is a sequence of DNA before the gene to which different kinds of molecules and enzymes and whatever combine, and that these are the ones that are going to transcribe the gene. Okay, this sequence is called a promoter. Of course, there is a lot of details that I'm so ashamed of doing this in front of all the audience in YouTube and Santiago. But anyway, I'm dry. But just have a little bit of a sequence of DNA that controls the production, and this is what we call the promoter. By the way, now that I do it here, I'm going to tell you that this allows us to do another type of monitoring. I'm not going to go into details much, but the other day you saw the protein fusion, in which you fuse the fluorescent protein to the protein of interest. Another thing that you can do is you can directly put the promoter controlling the expression of a fluorescent protein. Okay, this means that you are generating, if you put this in your cell, whenever the original gene is expressed, this case is what we call COMG or COMS. I will come back to these genes later because they have a special meaning for us. So these are genes that whenever these genes, the native genes COMG are expressed, then you're going to have this cyan fluorescent protein or yellow fluorescent protein. And then I use false colors, but terribly chosen for daltonism. I'm sorry for that, which are green and red. And then you can look, and then this can tell you that, look, in this cell, this gene is being produced because this promoter is being activated. In this cell, this gene is being produced. So you're going to see the rest of the day and tomorrow a lot of this. Because if we're looking at gene expression, I don't want to put a protein fusion because I don't want to alter the function of my protein. You remember that I had to tell you, I have to check that the fluorescent part is not affecting the function of the protein. If I do it like this, I don't care. Because then the native protein is the normal one. I don't touch it. I just add an extra copy of DNA to my thing, and then it's just a marker. Whenever the other protein is being produced, this one is also being produced, and hopefully has less effect. Okay, this is what's called a promoter fusion for obvious reasons, and I just wanted to tell you. Okay, now I have introduced the concept of a promoter. I was not planning to, but anyway, I hope it's okay. Again, as usual, please interrupt me because I may be saying completely crazy things or. So anyway, let's continue to where we were. So do the mapping. If this E is not the enzyme, but the promoter, the idea is that the promoter is going to bind S, which is a transcription factor, instead of having these things. When the transcription factor binds to the promoter, and it can bind and unbind, it's going to form a complex, and from time to time, this complex is going to turn on production, okay? And that's the assumption, and this is going to be the mRNA that I produce, and eventually the protein. Everything, is this clear, the mapping? Now, I don't know, did you ever see the maths behind the enzyme kinetics? Okay, I'm not going to go through the whole thing, just quickly, and since these slides are going to be there, if you want to take a look at it. But so first of all, we can write down an evolution. Oh, this is too early. Oh no, I think we can do it already because one thing that I haven't told you is that we are going to use the rules of the mass action kinetics. Chemical reactions, I hope we all know how this works. So we are using mass action kinetics. So whenever we have reactions here, this term here corresponds to the rate of production of this complex through this reaction here. That is why it's K1, this rate, multiplied by the concentrations of E and S. These are the K terms which correspond to these other reactions. We are losing ES through this reaction and through this reaction. So we can write down these equations using mass action kinetics. So far, so good for everybody, more or less? It's very simple. Okay, now the other thing is we assume that E is conserved. E was an enzyme in the case of enzyme kinetics. In the case of promoter, it's also conserved because the promoter is there. It's a piece of DNA that's there forever and ever. So there is it's conserved because the promoter is either free or bound to the transcription factor. So you have this conservation law. So you can put it here, and then you get only an equation for ES. And then you, assuming that you are in quasi-steady state, that the complex is all the time more or less in steady state, then you can get to the concentration of the complex. And if you have the concentration of the complex, just by multiplying by K2, you have the rate of production of P. So anyway, you can go later through it if you want. But at the end of the day, what we have is the rate of production of P is proportional to the concentration of the complex, which we obtained earlier here. I haven't gone through the details. I'm just going to highlight the result, which is something like this. Okay, now this constant Km, I just want to highlight this constant Km. First of all, it's the half maximal expression, right? You all agree? Because if the transcription factor is equal, concentration is equal to Km, this is S, we have beta over 2. So it's the concentration of transcription factor at which we have half maximal expression. So this is this point, okay? In the enzyme kinetics language, it would be the concentration of substrate at which I have half maximum production of my product. But anyway, so this is very relevant. And I just wanted to point out that this is a combination of all these rates. Okay, so it's a combination of the rate at which the transcription factor binds to the DNA and produces the protein, which are genetically controlled, these rates. Okay, and these constants here are controlled by the genome. This will come useful later. Okay, so anyway, this is the typical curve. And it's very reasonable. This is saying, okay, sorry, when you say here substrate, you can assume that this is your transcription factor. At the beginning, I increase my transcription factor and the production rate is more or less linear. You can do the limit, right? Very simple. In the case of my biology students, I go do it very detailed, but here I think I can go fast. At the beginning, this is approximately linear. And then due to the saturation term here, this S in the denominator, you're going to have a saturation. This curve, I'm telling you, it appears in all the books of enzyme. I mean, it's point number one in enzyme kinetics thing, and I'm telling you, all students in biology, I think more or less know, because that's the example that they have. Okay, so this is the first way in which we model this process, in which we call it transcriptional activation, because the transcription factor S binds to the promoter and produces the protein. Okay? That's point number one. Now, what I like to claim is that this is already nice, but it's not extremely. This thing is nice because we can activate the gene, but it's not very robust. You agree? And we are going to talk now in the next few minutes about the concepts of robustness and so forth. It's not very robust. I told you earlier that there's never zero in biology. There's never zero. So if I want to make sure that the gene is turned off, I should have zero concentration of transcription factor, but there's never zero. There's always some leakage in our promote, so there's always going to be some copies of the protein produced. So sometimes we will be here, and then as soon as we have a little bit of a peak in S, this is going to go up. So and this is linear here, so it goes very quick. It grows very quickly, if you will. I very much would like another type of activation curve. You can imagine, right? And I'm going to give you a solution, and the solution is here. The solution, I would like to have a curve like this, in which instead of the one that I just showed you, which is the one in blue, I would like to have something like the one in red, right? In which at the beginning nothing happens, and then we have a threshold. And when the concentration of transcription factor goes above that threshold, then we have a lot of production. It's almost all or none, right? All or none dynamics is very helpful even for us. Well, maybe for those of us interested in dynamical systems, it's not extremely nice because it leads to almost discontinuities and so forth, but thankfully it's not completely discontinuous and we can do approximations, right? So it's basically an all or none response, okay? And then we have this type of thing here. And how do we accomplish this? We accomplish this taking into account another concept that biologists know extremely well, which is called cooperativity. The typical example is hemoglobin in our blood, that you need 4 atoms of oxygen. I mean, when you have more atoms of oxygens, then eventually when you have 4 of them, then the molecule is taken up as much as possible very quickly. So having multiple copies of one object together enhances a lot the behavior. Now you can see it here because imagine that instead of only 1 copy of the transcription factor, you need 2 or 4, which is very common. Okay, so if I need, sometimes these transcription factors form tetramers, what they're called, okay? A molecule with 4 copies. And this is what binds to the promoter. So in a very simplistic way, and I'm not going to enter into this, because of course this is a very complicated reaction in itself. So first you would have the multimerization, the tetramerization, and then you will have the binding, but I'm ignoring this and putting everything together. I am assuming that you need n copies, and then when you have n copies, this is the complex, and then you will have the production. That's what's called cooperativity. If you do the math, you'll see that now the function is like this with an exponent here. And when the exponent is larger than 1, you start getting this type of behavior. And if it's very large, you get a very nice, sharp thing. And having thresholds, my claim is that cells having thresholds is extremely useful for cells, because now you can have switches that you can control and you have thresholds. Like you have in electronics, you have to go past a threshold and then something happens. Or neurons, right? But I'm not going to talk about neurons today. It's the same idea. If some of you are working a lot in artificial intelligence, you will recognize this type of things, because this type of sigmoidal curves, these are the ones that you use to represent the dynamics of neurons in your neural networks, and then this leads to deep learning and so forth. Okay, so but cells have this in a very natural manner. Now I expect a question from somebody. Oh, I have something in the chat. Oh no, Plickers is great. Thank you, Gautier. Yes. I can see Giuseppe is also listening from wherever he is. Anyway, I expect a question. Does somebody have a question saying, no, this cannot work? This is weird. You are selling us a motorbike. ¿No? Nos estás vendiendo la moto. You are selling us a motorbike because now doesn't somebody detect a problem or some possible concern here in this figure if we want to have this beautiful threshold devices in our cells? If I were you, well, maybe I'm not being fair by putting you on the spot, but if I were you, I would complain about the fact that the red corresponds to 20. Because you might be thinking, oh, come on, you just told us about 2 or 4 tetramers or dimers, and indeed that's very common, let's say quite common, that these transcription factors operate as dimers or tetramers. But I will not be able in my life to make you believe that you need 20 copies. Because this is already, then this is very complicated. The efficiency is going to be reduced if you need to form a 20-mer. So you might complain and saying this is not realistic. Maybe it's more realistic the green, or even a little bit yellow is 5. I should have put a 4 here. But I was thinking mathematically more than biologically, maybe. I don't know. Well, I don't know. So anyway, so you might be thinking, is it realistic 20? It doesn't seem like it. And this I spend, as blind know, in my courses, I spend 2 or 3 days, 2 or 3 classes, showing examples in ways in which cells increase this. Okay, there are examples of very simple, and I don't have time because we only have 3 classes. So there are quite a few examples in which by very simple mechanisms, one is called molecular titration, the other one is called zero-order ultrasensitivity, another one is simply a cascade. Maybe this one is easier for you to understand. If I have not one, but several of these things is cascade, as I move in the cascade, imagine I have a cascade of n equal 2's. Every time I go to the next, if you compose a green function on itself, maybe we can, sorry, I should, green function. No, well, sorry, I don't know how to say it, light green function, anyway. If you compose this function in green with itself multiple times, as you compose more and more, you can already imagine that this is going to be looking like this. So if you have multiple connections of pairs of genes and transcription factors in cascade, you're going to increase the cooperativity. Okay, and this is very common. And in fact, in cells, there are many cascades. But that's one of the many mechanisms. There are others, molecular titration, zero-order ultrasensitivity, other mechanisms through which effectively you increase this number. So the cell is full, in my opinion, well, we know, we can measure, the cell is full of these threshold-like devices. Okay, that's the claim that I wanted to make now. Now, this is for activation. But of course, we know that some genes, some proteins can repress. You remember we had the 2 things, we have the 2 signs, and we need the 2 signs. How do we do it? And you can repeat the whole calculation. Now the trick is that if the transcription factor is repressing, production comes from the free promoter. Do you agree with me? Only when the promoter is free, you're going to have production of the gene. So it's not ES. Before what we had is that this thing is the one producing the protein, because the promoter needed to be bound to a transcription factor, and this led to the expression. Now the promoter needs to be free, because if the transcription factor binds, it's going to prevent production. So the expression comes from a free promoter. That's why these are now the chemical reactions. It's different from Michaelis-Menten, but you can do the same analysis, and eventually what happens is that you get a similar thing, but with S in the denominator. And then now you have this hyperbolic function or whatever, I don't know if it's correct, but you have this kind of thing, which again, biologists maybe don't know so well, but these are also very common. And then you have this thing, and we have the same problem. This is not very robust, because as soon as I have a little bit of transcription factor, I'm bringing this thing down. And again, if you put cooperativity, you have an N here, and then you have a beautiful switch in the positive, in the other direction. Okay, These are called Hill functions by the guy who basically founded biophysics, and he also has very important contributions to biochemistry. Hill, I don't know the name, I didn't know him, so I don't know his first name. Anyway, sorry. So these are Hill functions, and this is so-called Hill coefficient. But of course, they are very normal functions. For us, they are very important, and they're clearly nonlinear, and these are the ones that they appear all the time in these models, because whenever you have some kind of regulation, it's very common that there's going to be some kind of cooperativity. Okay. Okay. So far, so good. So we have. Okay. So I'm going to continue. No, there is no break in one of these hour and a half. Shall I continue? Okay. Let me check the chat. Okay, because now this was very trivial. There is no circuit in here. I don't have a diagram, but A activates B, or A represses B. It's kind of sad because as a dynamical system, this is not very interesting because the degrees of freedom are separate. A goes by itself, and B is just affected by A. So there is not much going on. So it is or no. If I have two genes, A and B, A is activating B, you could say it's two-dimensional, yeah, but I mean, I solve first A, and then I solve B, so they are completely uncoupled, if you will. Sorry, I wouldn't say this, but there's no feedback, there's nothing. So my point is that, okay, let's make this circuit interesting. And I can make it interesting by adding, by keeping 1 degree of freedom. I don't even need two. I have only one again, and just saying, okay, I am going to consider that the gene inhibits itself. Okay? And the equation would be this, right? So the production term would be a Hill function in which the transcription factor is X itself. Everybody agrees with me? And then we have a decay, of course, because everything decays. No? Everything has a finite lifetime at the very least. There's dilution, there are proteases, whatever. So anyway, this is the model for this circuit. Okay, so we have here the production term, which is depends on X itself. And now we have a nice dynamical system with only 1 degree of freedom. Now you might be thinking, this is probably academic, but it's not true. Cells have huge numbers of this. If you look at how many direct negative feedbacks are in cells, genes that repress their own production, there are a lot. Okay, there are many more than expected by chance. This is what's called a new network motif, but I'm not going to enter into this. But I'm telling you, there are a lot. So the question is why? What is the usefulness of this circuit? Okay, it's there. It's realistic. So let's look at the behavior. Okay, so let's go to the blackboard. Okay, so now I'm going to use the blackboard for the first time. So I go here, and I'm going to sit because I don't have enough cable here. So I am going to sit. Let's analyze this thing. Let's draw the, sorry, I'm going to do this thing markdown, and then I'm going to go here, and I have a blackboard, right? So let's do the phase line analysis of this thing together. No? So let's do it together. So let me write it again. I have here my notes so that I don't. So let's see if this works. So I have here the equation, right? Let me remind you, this is what we have. You can see what I'm doing, right? Okay, this is cleaner. I love chalk, but I don't want chalk to mix with the electronics. So I decided to forego chalk. I mean, I would do maybe what Inma is doing, which is all the time with chalk, but not combined. I don't like it. It's so that's why I'm using this. I'm sorry. I think this is more physical and nicer, but anyway. So let me write this thing. Okay, this is the equation. Okay, so now, of course, what we can do, one nice thing about this is that I can just use a lot of colors. So I'm going to use the colors. Okay, so if I have the production and the decay terms, I don't need to spend too much time in going over this, right? So I can just draw the phase line, and then what I have is that we have on the one hand the production term is a repression, okay? One of these Hill functions that are repressed as a function of X. Do you agree? Please stop me if there is something wrong or something. And of course, the decay is this. No, so we have this, and then we have this. And this is the beta 1 plus X over K to the N. And of course, as you can see here, there is a crossing point which corresponds to a fixed point, which is stable, right? Because on the left-hand side here, I'm going to change the color of this thing. On the left-hand side here, I hope you can see. Maybe there's too much light. Is it okay? Okay. So I can switch. Which one is it? Persian. No, this one. Yum. Pantalla que no. Pantalla que no. Pantalla que no. No. I don't know. These are things that are controlling the. Okay, doesn't matter if you can see. Okay, so then let's leave it. If you want to see, thank you, Thomas. Don't worry for the moment. I'm going to continue. If you anyway, what I was saying is that on these hands here you have production winning over degradation and vice versa. So this is a stable fixed point. Now, thank you. So my claim for my biologist friends, I mean, for you, it's not so impressive because you know, but I think once you know something, you do not appreciate what you have. Okay, I'm telling you, they have been teaching you this for many years, people like mathematicians, and I think I didn't appreciate this until I had to teach it to biologists. Because I tell biologists, look, you don't even need to solve a differential equation, let alone it's a nonlinear thing. You don't even need to simulate in a computer, just plot two lines. So this, I mean, Thomas said the other day that he loves graphical methods. I think we use this trick, and I love them for the same reason. Okay, so that's my selling point to biologists, because this tells you again that there is one fixed point and only one fixed point forever and ever, whatever the values of the parameters. That's the main thing. So, which is not trivial at all. If you look at the equation, if I show you the equation, okay, how many fixed points there are? I don't know, because if I look at the zero, then I have a polynomial with degree 9 plus n plus 1. How many degrees? Well, draw the two things and just have only one solution forever and ever, as long as the parameters are positive. Okay, which is what happens in biology, right? So anyway, I just wanted to point out, maybe if I erase this for a minute, let me remind you about the other problem that we had before, for reasons that will come clear in a minute. I could do the same analysis. What we did earlier, I was plotting directly A minus gamma, but I could do the same thing I have done here. This is this, and just putting them like this. So then I have something like this, and then I have something like this. So this is A, and this is gamma x, and it's the same idea, right? Why am I doing it like this? Because now it's easier to compare. Now it's easier to compare because, well, now you see that again, in this system, you also have only one fixed point. But one of the other things that I like about these diagrams is that they tell us many things. For instance, one of the things they tell us is, I always like to point out, is that this difference is the speed at which the protein is being produced, right? Is the rate, if you will. No, so this is telling us that at the beginning, the rate is large. As the protein production, as the protein is being produced, the speed is decreasing linearly because this arrow is decreasing. In this particular case, you can see what happens. It decreases, but it decreases very slowly. Eventually, it reaches the fixed point at zero speed, right? So it's as if I go to the wall running, and when I get closer, I go closer and closer and closer, and I hit at zero speed. I am very slow. Okay, so this system is slow. I'm going to put it here. This system is slow. Again, I'm sorry if I'm telling you trivial things, but I'm always excited to explain these things. And the good thing about being a professor is that you always have new students, unless they fail. But since they don't fail much, but since you always have new students, they never know. So I get to explain it over and over again because I love this. Okay, so what I'm telling you is that this is slow. The one on the right is very slow. Now the one on the left is not slow because it's a slowing down. But suddenly, when it has to go and hit the fixed point, it still has non-zero velocity. It almost, okay, of course. In other words, the velocity decreases very quickly. So you can already tell, just without doing any analysis. Of course, we could calculate the characteristic times, and if you want, you can do it. No, because now that you know all the equation and you know all the techniques, I would encourage you to use all the techniques you have learned in the other courses in this school to analyze this equation. And you can look at the characteristic times and whatever you see, and you will compare the characteristic times, and you will see that this is very fast. Okay, this goes very fast. Now, no wonder that this negative feedback system is a typical system used for control. In control theory, the simplest control theory ever is a thermostat, which is a negative feedback. No, if I am too hot, I cool down the room. If I am too cold, I heat up the room. This is a negative feedback, a thermostat. So I always tell my students, this is like a thermostat in a cell, and cells need this thermostat. This has a name in biology. An engineer would call this control. A biologist would call this homeostasis. I should have maybe, okay, this is homeostasis. So if you ask why do cells have so many direct negative feedbacks, it's because their function is homeostasis. Okay, homeostasis. Okay, now is this really, of course, to be homeostatic, you need to be robust. So what I would like to do is analyze the robustness of these things. Because I haven't told you yet, and we will see tomorrow, there is a lot of noise. So in a population of cells, these parameters, beta, gamma, they're going to change a lot. They're going to change quite a bit because every cell has its own amount of RNA, of enzymes, polymerases. So some cells are going to produce genes faster than others. So some cells are going to have larger values of beta than others. Also, some cells are going to grow faster than others. You can see this you put a bunch of cells on a microscope, and you will see, you can track them, and they don't live for the same amount of time, which means the dilution rates are different. Some cells are growing faster than others, and therefore some cells have different values of gamma than others. Okay, so now if this has to be a homeostatic system, this should be robust, so that in a population of cells that have a variability in beta and gamma, for instance, the response is the same. In other words, the fixed point doesn't change much. And maybe, I don't know, maybe I'm doing something that you have already, I don't know if Imma, because I was not present, I looked at some of Imma's talk, but I didn't look at all of it. So I wouldn't want to repeat myself. So let me just very briefly talk about robustness, right? Robustness. So if I draw again the phase line and the production term and the expression term, okay, this is gamma x, and this is beta x, beta 1 divided by x divided by k to the n, and this is x. So what happens when beta changes? You can see immediately that when beta changes, when beta changes, the only thing that changes is the red line, and it changes in a very specific manner. It only changes like this. Oh my goodness, what happened? Oh my goodness. Okay, so it only changes like this. Does everybody agree? So the right-hand side basically doesn't change. I haven't told you this, and I should, and I apologize. This point here, is k. I haven't told you, sorry, the thresholds that I apologize, I forgot. The thresholds of these Hill functions, everybody, I hope is everybody agreeing with me? The thresholds of these Hill functions is k, because it's the value, no, when, oh, I have to, sorry, when, when x is equal to k, this is 1 plus 1, beta 2. Okay, so this is when everything is half. So k is the threshold. Okay, so this means that if I change beta but I don't change k, the threshold is still the same. So then this means that everybody agrees beta is only changing the top part, the one on the left. So of course, what happens in this particular case is that even if beta changes, the fixed point, which by the way, it is this one, it's more or less, so I'm going to use the notation. I don't know if you use this notation, the asterisk in mathematics. Sorry, this is used by, I think this is used in the book by Steve Strogatz, but I forgot. Anyway, I'm using the, I don't know how to refer to fixed points. Maybe we should have, anyway. So x star, okay? So the fixed point is here, and it's not changing much when beta changes quite a bit. So this means that it's very robust. Let me just again compare with the, and I'm not going to change colors because it takes forever. But if I change now I change A, now the fixed points change a lot. Do you agree the difference? The system on the left is very robust. I can have many different values of beta. The fixed point is always going to be close to K. The system on the right is not robust. I'm going to have different values of A, which was the expression rate in that case, and the fixed point changes basically linearly, of course. The fixed point changes linearly with the value of the parameter, okay? So that's the robustness. That's why this is a good homeostatic and control system, if you will. Now, I don't know, I don't think that engineers teach control theory using this type of approach, but I think it's very useful to think of it in this term, in terms of cells and whatever, and looking at this, but I may be wrong, okay? So another thing, very quickly. What happens if gamma changes? I mean, because you're going to have cells that grow at different rates, so the dilution is going to be varying. If this is degradation also, because maybe there are proteases that are destroying the protein, and then the number of proteases in different cells is different. So you're going to have different values of gamma in different cells. So this means that different cells are going to be like this, right? And again, the same thing happens. Of course, with us. Of course, if gamma becomes very large or very small, you enter into the other parts and then you're done. But with a certain, in a certain range, it doesn't matter. You change gamma, and the set point, the fixed point is always at the close to the thread to K. Okay. Again, if you do it like this, if here, nothing. It's terrible. Okay. Now maybe I expect some other complaint or not. I'm complaining. There's a time in which we can complain now. The system has 3 parameters, right? Apart from the Hill coefficient n, I'm not touching, I'm not talking, but there are 3 parameters: beta, gamma, and K. Immediately, you will complain, or you could complain and say, look, come on, this motorbike only has 1 wheel because the system is not robust to K. Because if I change K, now I have a problem, because of course the fixed point changes with K. So this system is robust with respect to 2 of the parameters, beta and gamma, and not with respect to K. Okay? But I have to tell you, I took this from the book by Uri Alon that I always cite. I like it because it's a nice argument, even though, of course, everything is subtle and just oversimplifying things. K, as I told you earlier, depends on the reaction rates. K1, K minus 1, K2, you remember? These reaction rates are basically controlled by the genome, because it's binding rates, right? These are binding rates between the transcription factor and the DNA. What determines the binding? And I haven't told you this, but I think I can tell you this now. What determines the binding? It's geometry, it's the fact that they fit well, the thermodynamics, right? And this depends on the specific sequence, the different parts of the molecule which are encoded by the DNA. Right? In the DNA, I haven't told you this, but I think it's clear, no? You have a set of bases, which are chemical entities, which are chemical objects, with different shapes and thermodynamic properties. So if the genetic sequence changes, the binding affinities are going to change. But basically, these are determined by the genetics. It's different from beta and gamma, because beta and gamma are going to depend on nutrients, and maybe when you have some nutrient or and it's going to depend on the gene being expressed, and we will see tomorrow that gene expression is very noisy. So it is normal. I mean, beta and gamma are very high level parameters. How many enzymes I have, how fast the cell is growing. These are very high level. They're going to be noisy. But K is not very high level. It's very fundamental. It depends on the reaction rates, which are controlled by the genome. So if you're on a bunch of cells that are identically genetically, it's very hard to think that K is going to be varying a lot. Okay? So the claim that I got from Uri Alon, this is not my idea, is that genetically, I mean, K is not very so much in a population of isogenic, so to say, cells with the same genome. Since the K is not varying so much, it doesn't really matter that the system is sensitive to K. And this is still useful. And again, I insist that, I mean, cells use it all the time. There are lots of direct negative feedback. There are lots of genes that they replace their own expression. Okay, so now in order to be quantitative, and I'm going to just give you this as a kind of a homework. Well, I have an exercise list that I have put up in the GitHub thing, and I changed it from yesterday, because yesterday, I mean, since I have seen that I'm delayed, so if you downloaded some exercise list from yesterday, scratch it and take it again, because I have changed it. Because I am giving you an exercise to quantify this thing. And since you are very apt at mathematics, I don't need to be very, I don't need to do it very, I just want to quantify, how do you quantify the sensitivity? Okay, so there is this quantity that it's called the sensitivity coefficient. Sensitivity coefficient, okay? Which we define as Xi, which is basically the derivative of the fixed point with respect to the parameter, whatever parameter there is, beta, gamma, or whatever. But to make it dimensionless, I just normalize. And this basically is a different, it's a logarithmic derivative or whatever, I don't know. So there is a little bit of math, so you can do the calculation. I'm giving you as an exercise this, which I'm supposed to do. So you can calculate this. It's the logarithmic derivative of the output with respect to the input. P is a parameter. Okay, I repeat, P could be beta, gamma, or K. And I would like to ask you to calculate the sensitivity coefficient. Sorry, this derivative, of course. Sorry, I should say, to be honest, I apologize. I should say, to be rigorous, right? I should say that this is calculated at the fixed point, right? Sorry. To be explicit, because the derivative of a constant, I apologize. I feel so embarrassed. Anyway, so you agree with what I'm saying? No, sorry. Is this correct what I'm saying? No, sorry. This is the real. How does it work? Now I'm embarrassed. Anyway, you know what I mean, right? Oops. Oh my goodness. You know what I mean. Shall I put here the star or not? Somebody can help me. Oh my goodness.
If you put the star,
yes.
You remove the button on the right side. It's okay because it's a star depends on the.
Okay.
On the other parameter.
Okay.
Yeah.
Okay. So anyway, you can do this calculation, and I'm going to ask you, and I'm going to give you my result. Now, this result could be wrong because I copied it very quickly from my notes. So I apologize in advance. Just take it. I'm giving you the result just that I have. Okay. And if you get another one, let's discuss it. That will be fun. If P is gamma, for instance, the corresponding sensitivity coefficient that I get is minus beta divided by gamma K N. Okay. If P corresponds to beta, the sensitivity coefficient that I have is plus beta gamma K. Sorry, this is approximate. Oh my goodness. This is approximated. These are approximate values, provided N is much larger than 1, by the way. I haven't told you this. You might be shouting inside, but I haven't told you. First of all, we do the approximation that N is much larger than 1. So it's basically a step function, almost. And then, of course, you obtain approximate forms because I mean, this is a nonlinear system, so we will never get such nice. So it's approximate. And if P is equal is K, then this is approximately equal to 1 plus 2 over N. These are the values that I got. They could be slightly wrong. Please double check if you will. And now what I would like to point out is, as you can see, for gamma and beta, the sensitivity increases. Sorry, the sensitivity increases. What am I saying? This is the sensitivity. Yeah. Sorry. This is the sensitivity. So the sensitivity decreases when N increases. If I have once N is large, the larger it is, the smaller the sensitivity, which means that the higher the robustness. Okay. So a very small value of this coefficient means a high robustness. We can use it in the opposite direction, but I'm not going to enter into this today. Okay. You could also use it to measure sensitivity itself. Here we are interested in robustness. So in some cases, it's interesting that C is very small. In some cases, it's interesting that C is very large. And cells, the beautiful thing is that cells have, of course, ways of doing both. And the Hill coefficient and the cooperativity is a way, as you can imagine, to increase the sensitivity a lot. But here we are interested in very small C, very small sensitivity, means slight robustness, and we get it the larger N is. Whereas in this particular case, the larger N is, the sensitivity goes to 1, because of course, we got a linear response. So this is already immediately telling us that this circuit is robust with respect to beta and gamma, but it's not robust with respect to K. Okay, just for fun, you can do these calculations. I think it might be reasonably fun. Okay, so far so good. Any question, comment, stupid things I have said, or let me look at the chat. Nothing in the chat. I hope the people online are not sleeping already. Here you cannot sleep because I'll see you, but no questions? Okay, let me go. This is a very simple CG circuit. I couldn't get any simpler. So I think given the time that the two-dimensional version I will do tomorrow, my goodness, okay, let me see if I can, because I haven't finished. I want to do the opposite version. I have done the negative feedback. And let's do the positive one. I haven't told you this, but I don't know, since a few years ago, by the way, since I entered into biology, I didn't realize the importance of feedbacks. I spent my whole career studying differential equations or partial differential equations in which we had feedback terms. And I just didn't think in terms of feedback. I don't know if this is the case with some of you, okay? When I started getting into this, then I realized, and now I see everything in terms of feedbacks. It's because having a feedback implies that now this determines the dimensionality of your dynamical system and the ability of the dynamical system to be dynamical, okay? So whatever is not connected via feedbacks, it's very trivial. It's somewhat trivial because you can solve in a hierarchical way. And I didn't realize this until I entered into biology. Maybe it was my fault. And I have to tell you, that's why I think, and I'm not going to go into this today. I gave a talk in the conference the other day about this, because in my opinion, feed-forward networks are very nice, and a lot of people are making a lot of money in Google, in Facebook, and whatever. But these feed-forward networks have limitations and are a bit boring from a dynamical systems point of view. The fun comes when you add feedbacks, because this is when you have dynamics and increase the real dimensionality of the system, okay? And that's what I explained. I don't know if you saw that, but that's because it's becoming my obsession little by little. Now I interpret these things in terms of feedback. That's why I don't want to get stuck here with negative feedback. I'm going to go now to positive feedback for just a few minutes, hopefully not too many, okay? So let's go to another one, another slide. And the idea is that now we go here. Oh, this is the next circuit. Trivial, one degree of freedom. It's still a genetic circuit with one feedback. This is much more fun than many feed-forward networks, and I apologize to everybody in the YouTube world that is a fan of artificial intelligence. I made fun of Google and Facebook in a screen. I should. Okay, sorry, Mark Zuckerberg and company. Okay, so. I have just one degree of freedom with feedback. This is already very interesting, in my opinion. Okay, so let's do it. So this is the system. So let's write down the equation together. So of course now the heel is going to be activating, and then we got this. And again, let's use for just one more time the color coding that we have here, and let's do it here. Okay. So here we have, again, the functions. So let's draw this one, and let's draw this one. And now immediately you can see something that probably you know already, but it's already very interesting, no? With this very simple circuit, which is now you have 3 crossing points between the production and the decay terms, which means that you have 3 fixed points, and these 2 happen to be stable, and this one happens to be unstable because the flows along the phase line go like this. Sorry, I hope I'm not being too. I'm just going to erase this. Oh my goodness. I'm going to regret this. No, I think I'm done. Right? So you have, immediately you see that there are parameter ranges in which here you have 2 fixed points that are stable. And I do not need to go into this. You know this very well. You can analyze it in detail. This is graphically, we can see the stability, but of course you can do the stability analysis, at least approximately, or whatever you want. But the important thing here is that now we can have multiple steady states for the same parameters. And now for cells, this is super important for 2 reasons, if you will, that are related. One is that with the same genome, you can have different types, which is the solution of the problem I told you yesterday, and now we can see it, right? If I need to have skin cells and blood cells using the same genome, of course, this is only one. This is an oversimplification, but just to show anybody that with only one feedback, positive feedback, you have bistability. So this gives to multistability. Another important thing, if you will, is that this leads to memory, right? And I forgot I wanted to show some synthetic biology papers in which they prove this synthetically. You can write memories onto cells because you can have cells in here. You bring them to this fixed point, and then you release the thing, and they stay here, right? So you can have memories. This is a 1-bit memory. So in other words, a positive feedback is a 1-bit memory. Now there are quite a bit of positive feedbacks in cells, but not so many as negative ones. Positive feedbacks are more common involving multiple proteins, multiple genes, for reasons that I don't have time to explain, and other kinds of functional uses. Okay. And the other thing, the last thing I wanted to tell about this, maybe I don't do so much math, and I think it's very important to be rigorous, as Tomás, Mayra, and Imma are also showing you. But I do it graphically. For instance, with this, I can graphically write down a bifurcation diagram, right? We can do it together. If I ask you to do the bifurcation diagram for which means plotting the fixed points with respect to beta, right? So let's do a bifurcation diagram, right? So first of all, when beta is equal to 0, which means that so in other words, let's do it like this. Sorry, let's do it like this. So you can see what happens, right? So when beta, so first of all, the fixed point at 0 is always, it's always stable. So this is stable all the time. This branch, you have a stable branch. And at some point, when you start increasing beta, at some point you have a saddle node, right? Well, I don't know if you already have looked very carefully into this, and I apologize to Imma because I'm sure that she has already explained. So you have a saddle node bifurcation, and then you have a saddle, or an unstable fixed point in this case, and a stable fixed point, which means that you're going to have something like this. By the way, the unstable fixed point is not going to change much. So I'm going to plot it like this, and the stable fixed point is going to do something like this, right? And this is a saddle node, right? Sn bifurcation. Okay, so just without doing any analysis, just by thinking a bit how this works, then we can see that we have emergence of bistability. No? Now, what I told you about the memory, the important thing that, I mean, for me, one important question, because now, for instance, imagine that I want to claim that this is a circuit for development, for the production of, for the transformation of one cell into another. Let's imagine the following. So imagine that this X determines that a cell is going from one state into another, from a stem cell to a certain progenitor of blood, whatever. Okay, so we need to increase X. So you might be thinking, look, I have a signal here, which are many that come from, they can be maternal signals, they can come from other cells. I mean, the world of decision making and development is amazing from a point of view, from the mathematical point of view. So I'm not going to enter into this. Imagine you have a signal beta, at the beginning is 0, the cell is pluripotent, is stem cell. Now you start getting beta, and at some point, you might be able to, at least the possibility for the cell to become active and become something else is there. Maybe it difficult. I mean, in this particular case, of course, since this is always stable, it's not so easy, but imagine there is a second perturbation that brings it up. Okay, for instance. The problem now comes, so imagine just a transient perturbation, a pulse of a signal. You are here, you have a pulse that brings this thing past the saddle point, and then you go up here. But now the problem is what happens with this beta. The signal that is the signal that makes the cell transform the state, if you bring it down again, because the cell doesn't want to be producing signals all the time to keep something in there, eventually you want to bring beta down. The problem with this is that when you bring beta down, the system goes along this line and then it falls, right? So this is not a very robust decision. So that's not very good for development. So I have an exercise for you. That's why I wanted to mention this, which is that there is a way of making this decision irreversible, which is by bringing this saddle node bifurcation to this side. You can change parameters or add things. So that now I bring this saddle node bifurcation to the negative part. I can bring this bifurcation. Sorry, and the people online don't see. I think they are seeing. I don't know if I don't know what they are seeing, but anyway, I'm just pointing it here. So I bring this bifurcation to the negative part. Because if I bring this bifurcation to the negative part, and now I have something like this, of course, I cannot reach the negative part. So this means that now, if I bring my point up here, and then I remove the beta, it's going to stay up there. This is an irreversible decision. This is perfect for development, because I only need the signal for a while, and then I bring it, okay? So I'm going to, there is an exercise that I put on that website to make this thing irreversible. And the way to do it, as I will show as you should see, you do it, okay? Is by adding what I could call basal expression. So in order to get, so first of all, I'm just going to label this thing. This is multistability. I'm going to be precise. And then I want irreversibility. So I want you to look at the exercises, and I want you to take a look at this model. What I'm adding here is this alpha parameter. Yes, this alpha parameter. Can you see? Blue is terrible, no? I'm adding here this alpha parameter. What is this alpha parameter? This alpha parameter can be interpreted, first of all, as a leak. I mean, it's an expression of the, it's a constant expression of the gene that happens without the transcription factor, or it could be another factor that is non-regulating. It's just that it's you have some production constitutively. Okay, so imagine that you have some constitutive production, and on top of that, you have the regulation. Okay, but imagine you have this parameter. Well, what I suggest that you do in the exercises that I just put in this is that if you do the same kind of analysis for alpha, you should get an irreversible thing. Mathematically, there's no difference between these two. And it's true, but it doesn't matter if it's in the negative side or in the positive side. But for cells, it is very important because I cannot access the negative part, because I cannot have negative concentration. So being in this concentration here makes the decision irreversible. And you can accomplish this in this case by playing with alpha. Okay? So I would ask you to do it if you want, to do the same analysis of this equation like this and just plot the bifurcation diagram, and you will see that this will resemble the situation here, which means that in this particular case, if you interpret this as a signal, the signal can induce cell phase change. And when you get the signal away, the cell gets stuck up there in the on state. So this means that the decision is irreversible. Another one of my heroes, which is Jim Ferrell, published a paper, I would say already almost 20 years ago in Science, in which basically not the same equation, but an equation like this. I mean, if you look at it, there's a paper that I'm not sure if it's freely available. I can put the link or something. It's a science paper, a paper in the Science Journal in which they have, of course, experiments, but they have a model, which is a one-dimensional ordinary differential equation, similar to this. And the idea is just this. Of course, there's a lot of work experimentally, but the idea of the paper is that you can get, this was in the frog embryo again, okay? This was development in the frog embryo. And it was showing that the circuit enables this type of irreversible decision. And this is one of my beloved papers, top papers of history, in the sense that I go back, and it's so simple and it's so elegant. It's so elegant, okay? And it underlies the existence of this kind of irreversible decisions, okay? Now, I feel a bit bad. Let me finish because I'm already past, I have taken all the time. So anyway, here, I'm going to leave this. This is just plotting the whole function. That's not as nice. This here, I'm plotting the whole function, but I think it's better to plot the production terms and the degradation terms. I'm a bit lazy, and I never upgraded this with the right thing. But if you plot F, the point here maybe is that if you plot F, you get the same result, but it's less intuitive, right? If you do positive feedback, you get this. Oh, I forgot. Okay, tomorrow, what I'll do, okay, this is the same thing. I'm plotting this thing instead of plotting the production and degradation separately. And these are the exercises. I have shamelessly put here a commercial for UPF. I'm sorry, but I use this to make it official. I don't know. So anyway, so these are the exercises, and these are the two ones that I just told you. Okay, that maybe you can try. If you want to send me the results, I mean, of course, we are not grading this thing, but if you want to, or are we? No, we're not grading this thing. So if you want to send me the results, just feel free to email me, and that's it. And then what we will do, again, let me remind you, this is where you get this thing. So tomorrow we will start our afternoon session with a Plickers question directly referring to this, to make some memory. I think it'll be good. So we start with the Plickers, and then we move on to finally to the finally we move on to two-dimensional, at least to have two genes. But already with only one gene, imagine that we can accomplish. Imagine that we have 20,000. That's my point. There is so much potential in a cell combinatorially that it's reasonable that they can do so many stuff. And dynamics is very important. Okay, so see you tomorrow then. I'm sorry that I'm already a little bit tight. So anyway, bye-bye everybody online. Okay, bye-bye. Okay, see you tomorrow. Well, I'll be here. Thank you. No. Well,